Reductionism is the scientific process of breaking down complex phenomena into smaller, more understandable components. This methodology has paved the way for remarkable breakthroughs, especially in physics and chemistry. Scientists often approach nanotechnology with the wrong assumption that small systems work like large machines, where everything is predictable and follows clear rules, similar to how a clock or car engine functions. But at the nanoscale, variability, randomness in movement, energy, and behaviour is not just noise, but a fundamental feature. For instance, molecular motors like ATP synthase, which power our cells, don't move smoothly like a car engine. Instead, they are constantly bumped by surrounding molecules and operate in random, chaotic environments. It's like trying to predict the exact path of a leaf floating in a turbulent river. Impossible with simple rules. While at first glance it might seem that these motors overcome variability and perform like large machines, the truth is they work precisely because they exploit the random fluctuations in their environment, like Brownian motion. Molecular motors harness the chaotic, non-equilibrium conditions to perform useful work, unlike traditional man-made machines that try to eliminate randomness for smoother, predictable performance. This is where reductive tools in nanotechnology fail, because they treat variability as a flaw or noise to be removed, while biological systems at the nanoscale rely on that variability to function. So, ATP synthase and similar molecular machines illustrate how current nanotechnology often misinterprets the role of variability, leading to less efficient or even non-functional designs. Similarly, trying to understand the origin of life by using straightforward chemistry fails because early molecules likely self-organised in unpredictable ways, much like how snowflakes form unique patterns from random water vapour. Even attempts to build synthetic cells struggle because natural cell membranes are flexible and adaptive, not rigid and predictable like man-made materials. In short, traditional linear approaches fall short at the nanoscale because they don't account for the random and dynamic nature of these tiny systems. While self-assembly allows molecules to spontaneously organise into structures, such as in DNA formation or membrane bilayers, these processes in biological systems are far more dynamic, adaptive, and nonlinear than simple self-assembly models can explain. For example, early life likely emerged through a series of highly unstable, far from equilibrium reactions, where random environmental factors like fluctuations in energy or molecular concentrations played a critical role. These systems didn't follow a simple blueprint, but evolved through a combination of randomness and selection, with emergent behaviours arising from the interplay of variables. Scientists using linear, predictable models often miss these subtleties. Similarly, synthetic cells face challenges because their components, such as membranes, need to mimic the adaptability of biological systems. Natural membranes can adjust their properties based on their environment, for example, becoming more fluid or rigid. But synthetic materials are often rigid and lack flexibility. Self-assembly may produce a membrane, but it doesn't necessarily recreate the adaptive, responsive behaviour seen in living cells, which is crucial for sustaining lifelike functions. The main issue is that scientists still apply reductive, classical thinking to these complex evolving systems. While self-assembly is a step in the right direction, it fails to fully capture the dynamic and emergent properties of biological systems, which thrive in unpredictable, fluctuating environments. This limits our ability to recreate truly functional nanomachines or synthetic life, as we haven't yet developed the tools to fully account for these random and non-linear behaviours. Just like a mechanic disassembling a car to find a fault, scientists use reductionism to understand atomic and subatomic particles, forming the backbone of discoveries in quantum mechanics and advanced materials. While reductionism excels in understanding fundamental particles, it struggles with complex systems like ecosystems or the human brain. These systems exhibit emergent behaviours that cannot be understood by simply studying their parts in isolation. For example, an ant colony's behaviour arises from interactions between thousands of ants, not just one. Chaos theory explores how small changes in the initial conditions of a system can result in vastly different outcomes. In nonlinear systems like weather patterns, minor variations can result in dramatically different weather outcomes, making long-term predictions incredibly difficult. This nonlinear progression and unpredictability underscore the limits of simple prediction models. In biological systems, variability is not just noise, but a key feature that allows organisms to adapt to changing environments. For example, our immune system uses variability to generate diverse responses to new pathogens. Much like a jazz band improvising its music, biological systems thrive on adaptability, turning variability into a strength. Fractals are structures that repeat themselves at different scales, a pattern found in many biological systems, such as blood vessels and neurons. These recursive patterns allow efficient function across various scales. A fractal-like structure in trees or lungs ensures efficient nutrient distribution or air exchange. The butterfly effect is a phenomenon where small changes in a system's initial conditions can have large and unpredictable consequences later. For instance, a tiny shift in wind speed can lead to a completely different weather forecast. This concept highlights the sensitivity of systems to initial conditions and the challenges of long-term prediction. In chaos theory, strange attractors describe how chaotic systems tend to follow certain paths, even if they don't repeat themselves exactly. Systems like a double pendulum exhibit seemingly random behaviour, but this randomness follows predictable patterns known as strange attractors. These patterns reveal an underlying structure in the chaos. Biological systems are dynamic and nonlinear, meaning they are constantly changing and adapting. Unlike mechanical systems that aim for stability, biological systems rely on feedback and variability to function. For example, the immune system adapts in real time to new pathogens, ensuring survival in a constantly changing environment. Emergence refers to complex behaviours arising from simple interactions. A single neuron doesn't possess intelligence, but when billions of neurons interact, they give rise to phenomena like consciousness. Similarly, simple organisms interacting can create ecosystems, demonstrating that the whole is often greater than the sum of its parts. Classical science, particularly reductionist methods, works well in systems that are stable and predictable, like planetary motion. However, in more complex systems like ecosystems or biological networks, classical methods fall short. These systems require more advanced tools that consider variability and emergent behaviours. Climate systems exhibit complexity through various feedback loops, teleconnections, and tipping points. Feedback loops like the ice albedo effect lead to further warming as ice melts, reducing reflectivity. Teleconnections, such as El Niño, show how localised climate changes can have global impacts. Critical tipping points, like the collapse of the Atlantic meridional overturning circulation, demonstrate the non-linear nature of climate change. Social systems showcase emergent properties from cultural evolution, economic patterns, social networks, and collective intelligence. Cultural norms arise from countless individual interactions, while social networks display the small world phenomenon, where simple rules give rise to complex behaviours. Similarly, collective intelligence emerges from group interactions, producing wisdom and problem-solving capabilities beyond individual knowledge. Modelling complex systems faces challenges such as computational limitations, data gaps, and interdependencies. Even with supercomputers, it's computationally intensive to model all interactions in systems like ecosystems. Incomplete data also makes accurate modelling difficult, and the intricate interdependencies complicate the isolation of variables, affecting model development and accuracy. Fractals play a critical role in human physiology, from the cardiovascular system to the respiratory system. The branching structure of blood vessels and the bronchial tree in the lungs exhibit fractal patterns that optimize efficiency. Similarly, the nervous system and bone structure demonstrate fractal properties, enhancing functions like information processing, strength, and resilience. The edge of chaos represents a balance between stability and flexibility, allowing systems to adapt optimally to changing conditions. Biological systems near the edge of chaos demonstrate greater evolutionary potential, while human cognition may leverage this zone for creative problem solving. Neural networks and cellular automata also show enhanced information processing at the edge of chaos. Nanotechnology aims for engineered precision, where structures are created at the molecular level with atomic scale accuracy. In contrast, biological systems thrive on variability, using stochastic processes to adapt and remain robust. Bridging this gap may involve incorporating biological variability principles into future nanotechnology for applications in medicine and environmental science. Quantum systems exhibit complexity through phenomena like superposition, entanglement, and decoherence. Particles can exist in multiple states simultaneously, introducing uncertainty. Entanglement creates non-local correlations between particles, adding to the complexity of quantum interactions. These emergent phenomena, such as superconductivity, arise from complex quantum behaviours. The future of complex systems, uncovering patterns beyond human comprehension. Integrating fields like network science and synthetic biology will push the boundaries of understanding, especially in areas like climate change and biological systems. As we manipulate complex systems, unpredictability raises questions about scientific responsibility and unintended consequences. Ethical considerations include equity, where interventions in areas like urban planning may disproportionately affect populations, and privacy concerns in data collection for social systems research. Acknowledging limits of prediction is crucial when policy decisions rely on complex models. Embracing complexity requires interdisciplinary collaboration, breaking down traditional academic silos. Moving beyond reductionism, scientists must adopt holistic approaches that account for emergent properties and nonlinear dynamics. Harnessing advanced computing and quantum technologies will be crucial in studying complex systems and embracing uncertainty as a fundamental part of nature.